Okay, are there any questions? If there's no questions, I'm going to begin. All right, can you see my screen?
Yes.
Great. Oops. Oh, good, that finished. All right, we'll do that later. That was the lecture just to finalize its video recording. So I can upload it now to the YouTube. So today we're covering Lab 9, the identification of gram-negative organisms. You're also working on your unknown work, and I do need to talk a little bit about that. I think I've got it here. If you scroll down to week 5 in the lab, you have the unknown report. Click on the link for unknown notes. In the unknown notes page, you should be recording your daily activities of what you're doing to identify your bacteria that I've emailed to you. And most of you have, in your unknown notes page, put up what your unknown number is, so good job there. But very few of you who put up your results for the gram stain, gelatin hydrolysis, and lactose fermentation. And I emailed those results to you, and you need to put those results in the unknown notes page, and then make your interpretation from those tests. And then look, once you've made your interpretations from those tests, look on the unknown list of possible species. I'll open that, to try and narrow down what your species is. This will be hard to see, but we'll live with it. And then determine which tests you want to run further. So far, only one student has asked for one test, and you guys do need to work on your unknown project, because this will take you time. You will need, on average, 2 to 3 weeks to get through this project. And that's because when you request one test, that test will be incubated for the appropriate time before I'm showing you the results, and then you'll write up your results and your interpretations, look at this chart, and then narrow down the species further, and then ask for another test. And so this process is going to take time. Are there any questions about any of that? My point is, you need to work on your unknown notes page and start narrowing down what species you have by working on your unknown project. Any questions about any of that? And when you request a test, just put it in a document. I don't have it. Instead of having a SpeedGrader and download submission, you will have a link for uploading a document. And just upload that document, and at the bottom of that document, put in what test you would like to request next. Do not put in a comment, because I put in comments for the results. And so if you have a comment in there, I'm just going to delete it. So put it in a document and then submit the document in your unknown notes page. You can submit an infinite number of documents to your unknown notes page. And that way everything will be together except for the first 2 emails I sent you. Okay? Any question about your unknown notes project? All right, let's move on. I said we were working on Lab 9, and you do need to work on your unknown work. And I realize a lot of you were working on your infectious disease paper until Saturday, so there wasn't much unknown work done before that. But now you should be working on your unknown work, and you must submit that by Saturday, 11:59 p.m. on June 12th. Okay? All right. So let's talk about Lab 9. That's the identification of gram-negative bacteria. There is some reading you'll need to do beforehand. The primary learning objection is to be able to interpret the results from the biochemical test, the IMVIC test, which IMVIC stands for indole, methyl red, Voges Proskure, and the citrate test, as well as be able to explain the carbohydrate fermentation test, the motility test, and the hydrogen sulfide production test. Be able to explain how to interpret triple sugar iron or TSI test, the SIMS deep, another test, and growth on McConkey's and hectone enteric or HE agar plates. Be able to use the biochemical analysis in this lab to solve the case studies involving gram-negative bacterial infections. Any question about the learning objectives? If not, let's move on. Members of the family Enterobacteriaceae are small gram-negative, non-spore-forming bacilli. They are all facultative anaerobes, and they all ferment the sugar glucose, and they're all oxidase negative. The Enterobacteriaceae have 53 genera, 24 of which are known to cause human infections. And these are some of the genera which cause human infections. Most of these genera are normal microbiota of the intestinal tract, meaning they are coliforms. They are opportunistic pathogens in that they can cause urinary tract and wound infections, pneumonia, and septicemia, as well as some of them can cause gastrointestinal, gastrointestinal itis. Common genera responsible for causing opportunistic infections include Escherichia coli, or E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera can cause gastroenteritis, and these include organisms in the genera Salmonella, Shigella, and then E. coli, Serovar 0157H7. Any question about the Enterobacteriaceae? We're also going to talk about Pseudomonas, which is similar to the family Enterobacteriaceae, but the Pseudomonas are in their own family, the Pseudomonaceae, and we're going to specifically talk about Pseudomonas aeruginosa, in that they are gram-negative bacilli. However, these bacteria are different in that none of them can ferment glucose, and they're all oxidase positive. Pseudomonas aeruginosa is an opportunistic pathogen causing infections, particularly in immunosuppressed individuals and burn and trauma patients. Pseudomonas aeruginosa is a healthcare acquired infection or a nosocomial infection. Excuse me, I want to sneeze and I don't want you guys to pick that up. Well, I never sneezed, and hopefully that won't happen. It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on gram-negative bacteria or gram-negative bacilli that are cultivated from clinical specimens. In this lab, we're going to talk about the common biochemical tests which are used to identify gram-negative bacilli. Some of the biochemical tests include the indole test, which is a test where organisms which produce the enzyme tryptophanase convert the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added, which reacts to indole to produce a red color at the surface. So tryptophan, that amino acid, in the presence of tryptophanase, produces indole, which when you add Kovacs reagent, changes the color from yellow to reddish. And normally that's a red color. For some reason in this picture, it is closer to purple. But normally it's reddish. Methyl red and Voges Proskure tests are two tests. Where's my... Are you guys seeing my screen?
Yes.
I don't see the picture showing me mine. So hopefully you can see Voges Proskure is two tests, which are then separated into two one tests, meaning Voges or MR methyl red Voges Proskure test is set up as one test, which is then split into two tests, the methyl red or MR test and the Voges Proskure or the VP test. And these tests are used to look at glucose metabolism and glycolysis, and you can have acid products being made or neutral products. The methyl red test detects the acid products, while the VP test detects the neutral products. So glucose and glycolysis can make either acid products or neutral products, or maybe both. And the MR test can find the acid products, and the VP test can find the neutral products of glycolysis. Where is my, I seem to have lost, oh, there it is, the mouse. So you set up MRVP test as one tube, and you incubate it for 2 to 5 days at 35 degrees. And then you split that one test into 2 tubes, one tube to test for methyl red. And if the bacteria produce acid products, when you add methyl red to the test, the acid products will keep the tube red, and then that tube will be positive for the methyl red test. In the VP test, you take the MRVP test and split it into 2, 1/2 going to the VP test. You add Barrett's A and Barrett's B. Shake it to get oxygen, and then after about 60 minutes, you look for the production of acetone. If acetone is there, the VP test will turn red, and then that will be positive for the VP test. If acetone is not there, the VP test will remain yellow, and that's negative. So VP negative, VP positive. For the MR test, it'll be red if it's MR positive. If it's MR negative, it'll be yellow. Any question about the MR test or the VP test? All right, moving on. Citrate utilization test. This is a test to see if the bacteria can utilize citrate as its sole carbon source. If the bacteria can metabolize citrate, it'll grow on the tube and use nitrogen. And if nitrogen is used, it will change the color of bromothymol blue, making it more basic. And so if citrate is used, citrate is not used, the citrate test will remain green, so that's negative. But if the bacteria can metabolize citrate, they will then use nitrogen, and the tube will become basic. The pH will, would that increase? And that will be a positive citrate where the tube turns blue. Any questions about citrate? If not, let's move on to carbohydrate metabolism. We're looking here at the fermentation of a single sugar. One sugar is put into a tube, and that sugar could be glucose, lactose, sucrose, maltose, or some other sugar. If the sugar is not fermented, the pH will not change or will not become acidic, and so the tube will remain reddish or pinkish. If the sugar is fermented, that will produce acid products, and the tube will change color to yellow because of the acid changing the pH indicator, phenol red. If the sugar is fermented, it may produce gas, in which case you'll see a bubble in the inverted Durham tube. So this is negative for fermentation. This is an A for acid in fermentation, and this tube is AG for acid and gas in fermentation. And that's how you score the carbohydrate metabolism test. Moving on to motility, some bacteria produce flagella, and if they have a flagella, they can move in the motility media. The motility media is a very low concentration of agar, and it is a semi-fluid media, meaning it's not solid, it's not liquid, it's semi-fluid. And if the bacteria can move, you will see the bacteria move out of the stab, and the media will be hazy around the stab, or in this case it's hazy everywhere because the bacteria are very motile. If the bacteria are non-motile, they will only grow in the stab, and so you'll see a very visible stab mark in the tube. And that's the motility test. In the hydrogen sulfide production test, we're looking to see if the bacteria can produce hydrogen sulfide. If they can, they will change the test to blackish or black. You can test for hydrogen sulfide production by more than one test. You can test for it in the triple iron, that should be triple sugar iron, I don't know how that got reversed, and TSI, or in the sulfide endomotility test, and that's the SEM agar test, or on the hactone enteric agar, or the HE agar plate. All three of these tests can test for the production of hydrogen sulfide, and they'll all turn blackish if hydrogen sulfide is produced. So this looks like it's a SEMS tube, and it turns blackish when hydrogen sulfide is produced. It does not change color or is not black if hydrogen sulfide is not produced. Any questions about hydrogen sulfide? If not, let's move on to the triple sugar iron agar slant, or the TSI slant. This test can be used to test for the production of hydrogen sulfide, but it is also looking at the fermentation of sugar in both the butt, meaning somewhat anaerobically or anaerobically, as well as the fermentation of sugars on the slant, and that would be aerobically. In the TSI tube, it has only 0.1% glucose and 1% lactose and 1% sucrose. And so it has tenfold higher lactose and sucrose than it does have glucose. And it has the pH indicator phenol red. An inoculation needle is used for inoculating a TSI tube. First you get bacteria on the needle, and then you stab the needle into the butt, and you should go down about 2/3 of the way from the bottom of the slant to make sure you get the bacteria in anaerobic conditions, because near the top of the butt it won't really be anaerobic, and obviously on the slant. And then you take the slant and pick up bacteria, and then you inoculate bacteria on the slant so that we have bacteria growing in both the butt and on the slant. you look at the tubes and compare it to a table. If the tube is unchanged, then no sugars were fermented, no gas was produced, and no hydrogen sulfide was produced. If hydrogen sulfide is produced, the bottom of the tube will be black. It'll only be on the bottom, not on the slant, because hydrogen sulfide is only produced under anaerobic conditions. You can't see the color of the tube if it is reddish or yellow because the black is hiding it, but if it is black, you can assume that the sugar has been fermented and that the color would be yellow. And that's just a trick you need to know about the TSI tube. So tube 2, a sugar is fermented because it's black. However, the slant is reddish, and if the slant is reddish and the butt is yellowish, it means that only glucose is fermented. You don't need to know this, but the reason why the slant turned red, I'll go to this tube, and the butt stayed yellow is because only glucose is being fermented, and glucose is at limiting supply in the tube. Aerobically, all of the glucose gets used up, and so the bacteria will metabolize the proteins, which makes ammonia, which is basic. So initially the slant was yellow from fermenting glucose, but later on all the glucose gets used up, and then the ammonia gets starting to produce, and it'll change the color of the slant from yellow to red. And that's why the slant is red, if glucose is the only sugar fermented. The butt remains yellow because the metabolism of glucose happens slower anaerobically, and so glucose is still present in the butt. So if the butt is yellow and the slant is red, then only glucose is fermented. If, on the other hand, the slant is yellow and the butt is yellow, then we know that both glucose is metabolized and at least one or both of the other sugars. We don't know which sugar is metabolized, but at least lactose or sucrose is fermented, and that is why the slant is yellow. And maybe both of them will be fermented. Glucose is the base sugar, so you can assume that glucose is fermented. In fact, lactose and sucrose will be converted into glucose and then fermented. Okay? If gas is produced, you'll see a bubble and cracking of the agar. So in tube 4, we have glucose and lactose and or sucrose fermented, gas produced, and no hydrogen sulfide produced because there's no blackening of the bottom of the tube. All right, TSI is complex, but I will always give you a table to help you explain the results to the TSI tube. Any questions about that? Okay. If there's no questions, let's talk about the sulfide endomotility test, or the SEM-Agar-DEEP. SEM-Agar is inoculated by stabbing the center with an inoculating needle, and then incubating it, and then evaluating the growth of the bacteria in the SIMS tube after you've added Kovacs reagent to the tube. Kovacs reagent is looking for the formation of indole, and if indole is there, the top of the tube where the Kovacs reagent is will be pinkish or reddish. And so tube 1, tube 2, tube 3, and tube 4 is indole positive. You also look at the tube, and this will be hard to see. Let me see if I can blow this up. And look if the bacteria is only growing in the stab. I don't even see the stab in the control tube. If the bacteria is only growing in the stab, or like in tube 4, you can see that the bacteria has diffused away from the stab. So tube 2 and tube 5, and I think tube 3, the bacteria is non-motile and is only growing in the stab. Let me make sure about that. Motility negative. Motility negative. Okay, I got it right. Tube 4, you can see easily, is motility positive. On tube 1, it's all black, but it is, what do you call that, diffuse everywhere, and so the bacteria have migrated everywhere, so tube 1 is also motile. Any question about any of that? Now, the third test is a hydrogen sulfide production, and you can see tube 1 turn black at the entire bottom of the tube. So tube 1, this species, is producing hydrogen sulfide gas. The control tube, tubes 2, 3, 4, and 5, are not producing hydrogen sulfide. And so the SIMS test is looking at 3 tests in one, tube or test. Any questions about SIM? All right, looking at MacConkey's and Hectone enteric agar, or HE agar. Both MacConkey's, meaning Mac, and HE agar are selective for gram-negative bacteria, especially the enterics. So if there's growth on the plate, you can say it's probably gram-negative. If there's no growth on the plate, you would say it's probably gram-positive. If there's growth on the plate, you can separate the lactose fermenting species from the non-lactose fermenting species by the color of the plate. If the bacteria look pinkish on MacConkey's, then the bacteria ferment the sugar lactose. If the bacteria are not pinkish, then they do not ferment the sugar lactose. Now on HE, it's similar, except the bacteria are salmon colored if they ferment the sugar lactose, and they are non-salmon colored if they do not ferment the sugar lactose. HE test can also look for the formation of hydrogen sulfide gas, and if hydrogen sulfide is produced, you'll see black colonies on HE. So hydrogen sulfide is produced by this species, so the colonies are black. This species is not black, And of course, both of them do not produce the sugar lactose. All right, any question about any of those tests? If not, let's talk about the Lab 9 exercises. You're not physically going to be doing any of the biochemical tests, but you are responsible for knowing and understand how to perform and interpret them. So view the video clips for carbohydrate fermentation, the SEMS test, the MRVP test, the triple sugar iron test, and the McConkey-Auger test. Also view some of the website on Microbuzz, and you can click this link to get to Microbuzz. Click on the differential test. It's a tab to the left of the screen. Expand the menu. You only need to click on the tests that you need to complete for step 1 in the worksheet of Lab 9. I don't have the worksheet here, so let me call up the worksheet. All right, so here's the table. You need to fill in the positive result, and the first one has been done for you. The cells or a spot turns blue when it is oxidase positive. You need to put in the negative result, which is pretty easy. The spot does not turn blue, but look up what the positive result is for indole. It's in the lab manual, but you can find it from, is it Microbuzz? You can also find it on Microbuzz. But the point is, you do need to click on the differential test of Microbuzz to help fill in this table. Okay? Any question about that? And in the worksheet, you got to complete the following questions. And then you do the case studies. Use this table to identify which species is causing the infection in the case study. And for the case study, you're going to be looking at test results. Fill in this table for the different test results. And then once you have the test results to the case studies, compare it to the table above, and then figure out the species which is causing the infection in case study number 1. And then do that for case study number 2, 3, 4, and 5. And let me see if I finished that. Here are the case studies. And you don't have to click on 5 different links. I've got them all together in this one file. So read through the case studies. It's showing your results here. There's the gram stain. Growing on MAC, and it's pink. You should know what that means. On day 2, you're looking at the different tests. Oh, sorry, this table got split. This is the SEMS test, the MR test, the VP test, the citrate test, and the TSI test. Hopefully you'll figure that out. And then put the results in the worksheet. Right here, and then use the table to identify the species which is causing the infection. All right, any question about the Lab 9? If there's no questions, I'm going to let you work on the lab. I will be here until 7:30 to answer questions. All right? Don't forget to work on your unknown project. So I'm going to stop the share and stop the recording.